of the VHL gene. In these families, they've been born with a slightly defective copy of the VHL gene from both mom and from dad. So it's a somewhat different situation, and we'll return to that in a moment. Other families have loss of function mutations in the EGLN1 oxygen sensing enzyme I just introduced to you, and yet other families have gain of function mutations of HIF2 alpha. They have mutations that make HIF2 alpha work too well. And so this provides genetic validation that this is the oxygen sensing pathway in man, at least with respect to the control of red blood cell production. Now, it might be a question to you, why don't these people with VHL mutations develop, for example, kidney cancer or hemangioblastomas? Why do they primarily develop excess red blood cells? And the answer is summarized on this slide, or at least we think this is the answer. So in kidney cancer, again, I've told you, you have a cell which is effectively devoid of the VHL gene, and it's making much too much HIF, shown here in red. But the normal cells are producing normal amounts of HIF, because in the case of sporadic kidney cancer, the host has 2 normal copies of the VHL gene. And even in VHL disease, there's at least 1 normal copy of the VHL gene in the remaining cells. And I've already told you, you only need 1 normal copy of the VHL gene to maintain normal function. In contrast, in these familial polycythemia syndromes, every cell in the body has slightly too much HIF because of the genetics I just described to you. So for example, this is the classical genotype of a family that has familial polycythemia linked to VHL mutations. They have 2 slightly defective copies of the VHL gene. So in summary, this is a big defect in HIF, but multiplied by a small number of cells. This is a subtle defect in HIF, but it's shared by every cell in the body, including every cell in the body that could make erythropoietin. Now, I want to return to the role of HIF in kidney cancer. We had shown many years ago, if you restore the function of the VHL protein in a renal carcinoma cell that lacks a normal copy of the VHL gene, those cells will no longer form tumors if put into an immunocompromised mouse. And so the question was, can we really link this to HIF deregulation or not? So Keiichi Kondo in the lab took these cells and introduced into these cells a form of HIF2 alpha that can't be recognized by VHL because the prolyl hydroxylation site has been eliminated. And now, once again, these cells could form tumors. Conversely, if he took these cells and eliminated the HIF2 alpha in these cells using so-called short hairpin RNA technology, these cells lost the ability to form tumors. Now, what's not shown here is this is specific to HIF2. HIF1 doesn't work here. So even though HIF1 is the more famous member of the family, even though HIF1 is frankly the one that appears in a lot of textbooks and sometimes in glossy ads and New England Journal article, magazines, it's really HIF2 here that's the problem. It's not the better studied member, HIF1. And this is summarizing in a cartoon form the fact that if anything in the laboratory, it looks like HIF1 is serving to constrain kidney cancer growth, and HIF2 is really the bad guy. And in fact, in many kidney cancers, we see that the production of HIF1 has been eliminated. And go back to one of my earlier slides, I told you HIF1 was on a chromosome that's frequently lost in kidney cancer. And we think this is one of the ways that kidney cancers get rid of HIF1 because they really want HIF2. So what are we going to do about this therapeutically? Well, for sure, we know that HIF is a master regulator of a number of factors, including VEGF that I've shown you here in red. We certainly believe that's why the tumors are so rich in blood vessels. Moreover, if you actually measure VEGF levels across a variety of tumor types, as was done here by scientists at Genentech, so on the y-axis is a measure of VEGF production. On the x-axis are the names of various tissues. The VEGF levels for the normal tissues are shown in green dots, and the VEGF levels for the tumors arising in those tissues are shown in red dots. And I think you can see that kidney cancer is the champion when it comes to production of VEGF. And we believe that's because of the biology I've described to you today. So this led early on to clinical trials of VEGF inhibitors in kidney cancer. These were early data from a randomized phase 2 trial where patients with metastatic kidney cancer were randomized to a placebo or low-dose Avastin, an antibody against VEGF, or high-dose Avastin. Each patient's tumor burden was normalized to 100% initially, and then the change in tumor burden was monitored over time. Parenthetically, this is a reminder that in the mid-90s, the medical treatment of kidney cancer was so hopeless, you could justify randomizing patients to a placebo. Fortunately, that's no longer the case, but it was true in the 90s. So you can see with the placebo, virtually all the patients had tumor progression. With the low dose, you can start to see some control, some stable disease. And at the high dose, some patients even went on to have actual regressions of their tumors. And this has been confirmed now in multiple phase 3 trials where patients with kidney cancer randomized to VEGF inhibitors such as sunitinib or sorafenib did much better than patients, for example, treated with a placebo or a standard of care interferon alpha. So the good news is that the drugs have an effect. And parenthetically, I think every clinician knows at least some kidney cancer patients who have seemingly derived a lot of benefit from these therapies and have sometimes been on these therapies for several years or more. But as Javier pointed out, we have to do better because virtually all patients eventually relapse, as shown here. So where else can we go? Where else can we look? Well, many years ago, in collaboration with Rick Klausner's group and George Vanderwood, we showed that VHL minus renocarcinomas are hypersensitive to the ligand for the MET tyrosine kinase called HGF. And I should point out this was really work of Rick and George with minor collaborations from our group. And so this suggested there might be some crosstalk between the HIF transcription factor and the MET receptor tyrosine kinase. Moreover, we had done a study where we systematically inactivated various kinases using genetic tools in VHL defective renal carcinoma cells or isogenic cells in which we had restored the function of VHL. And we had discovered that VHL minus cells are very, very sensitive to loss of MET signaling. And this is VHL dependent because when we restored the function of VHL, now the cells seemingly were less dependent on MET. And so for this and many other reasons, we've wondered whether MET might be another good target in clear cell renal cancer, just as it is suspected of being important in certain papillary cancers. And so recently, data were presented with a drug called cabozantinib that actually does inhibit both the VEGF receptor, but perhaps fortuitously also inhibits the MET receptor tyrosine kinase. These were patients with kidney cancer who had failed multiple lines of therapy, and in this trial, cabozantinib performed better than the comparator, which was everolimus. So I think this is encouraging, but I will also admit, as a scientist, this doesn't yet directly address the importance of MET, because you could argue maybe this is simply a better VEGF inhibitor than the other available agents, but it's at least consistent with, but does not prove that there's a role for MET. So there are now a number of these VEGF inhibitors approved for kidney cancer, and I think more are yet on the way. You may also know there's a role for mTOR inhibitors in kidney cancer, but I have to say we still don't completely understand why these drugs work in kidney cancer. One initial thought is depicted here. It's very clear that mTOR does play a role downstream of VEGF in endothelial cells, and so one initial thought was that these drugs were working in part by providing another way of shutting off VEGF. But I think there's more compelling evidence now that these drugs have a direct effect on the kidney cancer cell itself, especially when those kidney cancers have mutations in either mTOR itself or upstream regulators such as TSC. So I think we're beginning to understand which subsets of clear cell kidney cancer might be the ones most likely to benefit from drugs such as everolimus and temsirolimus. Now, what about immunotherapy? I suspect it's the same here, but in the United States, you read constantly about the promise of immunotherapy. I think in kidney cancer, so far, it's a bit of a mixed bag, so to speak. So there was a recent trial comparing an immunotherapy drug, nivolumab, a so-called anti-PD-1 antibody, against the standard of care in advanced renal cell carcinoma. I think if you look at the overall populations, I should also point out this was a randomized trial in patients who had failed at least one line of therapy. I think there was a statistically significant improvement in progression-free survival and in, and in overall survival, but I would be the first to admit that the benefits looked at the level of the entire population are rather modest. But I think buried within these data are a small number of patients who seemingly have obtained much more benefit. So once again, I think we have to begin to understand which patients are most likely to benefit from these agents and which are not. Now, I want to return to the story of HIF-2 alpha. So again, in kidney cancer, we think the real problem is HIF-2 alpha itself. Classical teaching would be that HIF-2 alpha is not druggable, however. It's in a family of proteins that historically have been difficult to inhibit with drug-like chemicals. But fortunately for us, two scientists in Texas, Rick Bruick and Kevin Gardner, identified a potential pocket within a particular region of HIF-2 alpha, and they realized that this pocket was big enough to potentially accommodate a drug. They then did a screen for chemicals that could bind to HIF-2 alpha and could inactivate its function, and they identified a number of chemical scaffolds that could do so. Now, my friends who are true medicinal chemists, that is to say, people who work in real pharmaceutical companies and not in academic laboratories, would say that this chemical was publication grade, but not pharmaceutical grade. This was not a good enough chemical to really start to give to people for many, many reasons. So fortunately, these scaffolds were out-licensed to a company called Peloton that has true professional medicinal chemists, and they optimized this chemical matter further, and better still, they allowed us to do some preclinical testing with this molecule. So the first thing we showed was that, as advertised, this chemical is a fairly potent inhibitor of HIF-2 alpha. So what you're looking at here are the mRNA levels for HIF-2 alpha itself, as well as 5 mRNAs that are regulated by HIF. And you can see at high nanomolar and certainly by low micromolar concentrations, you shut off the accumulation of these mRNAs. And this is really exquisitely specific because BNIP3 is a HIF target, but it can only be activated by HIF1. It can't be activated by HIF2. So you can see this is quite specific. We then created mice where we could monitor the activity of this drug non-invasively. And the way this was done was to create mice that had kidney cancers growing in each kidney, left and right, but where on the left the tumor had that firefly luciferase protein under the control of HIF itself, and so for shorthand we call that HRE. And on the right kidney, the luciferase was under the control of a workhorse element that doesn't care about HIF. And the animals were then treated with either a vehicle or the drug, and the animals were imaged pre and post treatment with the drug. And in every case, you can see that the HIF responsive signal goes down with the treatment with PT2399, and this is simply quantified here. So that said, for the first time, we had a drug that was actually bioavailable in a mammal and could shut down HIF2 alpha. And if you really want to get fancy, you can do the same experiment, but now treat the animals. You can watch the signal go down after you treat the animals. You can then wash the drug away and watch the signal return, and then treat them once more and turn the signal back off. So does this translate into an anti-tumor effect? The answer was yes. So here you're looking at mice that once again have tumors growing in their kidneys formed by VHL null renal cancer cell lines. Here the luciferase is always under the control of the workhorse element that doesn't respond to HIF. And so we're simply going to use light here to monitor the growth of the tumors. And so the punchline is shown over here on the right. In the animals that got the vehicle or the placebo, the tumors kept growing, whereas in the animals that got the active agent, the HIF2 alpha inhibitor, the tumors either stop growing or they actually start to shrink, such as had happened in this animal here. Now, is this also true in the metastatic setting? So we switched to a model where now the animals have lung disease. They have lung metastases formed by a VHL defective renal carcinoma line. And you can see once again that if the animals were treated with the vehicle, the tumors keep growing. You can see more and more light being emitted. We confirmed at autopsy that this is because of tumor growth. Whereas if you treat the animals with the inhibitor, now the tumors begin to regress. And this translates into a marked improvement in survival. So we're very excited about this compound. It's actually entered phase 2 clinical testing for metastatic kidney cancer in the United States, and is about to enter early stage testing for VHL disease. But there is one fly in the ointment, and that is, up to now, you could say, how do you really know the anti-tumor effects of this drug are really due to blocking HIF2 alpha, and not due to some other activity of the drug that perhaps was unintended, but makes it a toxic molecule that simply kills kidney cancer cells? So how would you formally prove that everything I just showed you is specifically due to blocking HIF2 alpha? So there's a trick that scientists can use here to answer that question. And so the basic question I just framed for you is, are these effects of this novel new drug really on target? Are the anti-tumor effects of this drug really specifically linked to blocking HIF2 alpha activity? So the trick here is to engineer a HIF2 alpha that by virtue of a single amino acid change will no longer bind the drug. So I showed you the drug binding pocket earlier, but with the help of Rick Bruick and Kevin Garner, we designed a variant where we changed a single amino acid in HIF2 alpha, and when you do this, now the drug can't bind to the pocket. So in theory, this HIF2 alpha should no longer be sensitive to the drug. That's exactly what we saw. So we used a new genetic trick called CRISPR to eliminate the normal or endogenous version of HIF2 alpha in renal cancer cell lines, and then replace the missing HIF2 alpha with a exogenous version of either wild-type HIF-2 alpha or now this HIF-2 alpha variant that's been engineered to no longer bind the drug. And when you do that, you see two things. First of all, in the cells expressing the drug-resistant HIF-2 alpha, now the drug really can't downregulate those HIF-responsive mRNAs compared to the cells expressing normal HIF-2 alpha, where you once again see in blue that the drug downregulates HIF-responsive transcripts. And moreover, if you treat cells, renal carcinoma cells, forming these so-called soft agar colonies with the HIF-2 alpha inhibitor, you can inhibit the soft agar growth of these cells. But again, the question is this simply non-specific toxicity? But the answer is clearly no, because if the cells have the drug-resistant HIF-2 alpha, now they're insensitive to the drug. And finally, we can reverse the anti-tumor effects of the drug in the cells that are expressing this variant. So as I showed you earlier, if you have metastatic disease and you treat with the HIF-2 alpha inhibitor, you cause regressions of the disease, but that's not true if the cells have the drug-resistant variant. So we now know that these effects are all on target. I should point out that for reasons we only partially understand, some VHL defective kidney cancer cell lines are very sensitive to this drug, and some are not. And we're beginning to understand that, but I think that's important to recognize that there may be some heterogeneity in terms of responsiveness to this drug. Finally, I just want to make one editorial comment. This, in my world of medical oncology and cancer pharmacology, is one of the most famous images that's frequently shown at meetings. This is a patient with metastatic melanoma who has a mutation in a target called RAF, specifically BRAF. And what we've learned over the past couple of years is if you treat such patients with potent drugs that can inhibit BRAF, you can induce Lazarus-like responses. So here this patient was near death with multiple cutaneous, and I will tell you, visceral nodules. And after starting a BRAF inhibitor, they look like this. Miracle. That's what we want to do. But the problem is, in 2 or 3 months, he looked like that again. So this underscores the problem of acquired resistance when you use drugs to treat cancer. And the question is, can we ever get over this problem? Because if we can't get over this problem, we're going to continue to show Kaplan-Meier curves, like I showed you earlier, where inevitably the patients are all failing. How are we going to go from that to actually curing people? So the good news is, all we have to do, I would propose, is relearn something that was learned a long time ago, and that is the magic of combination therapy. So imagine you have a cubic centimeter tumor that's going to already have about 10 to the 9 cells. Most patients in the clinic have 10 to the 10th to 10 to the 12th cells. So if you have a really good drug, but 1 in a million cells are inherently resistant to your drug, and you use that drug as a single agent, that drug will ultimately fail. You will be met with resistance. Your chance of cure using a single agent is approximately 0. But what we learned decades ago was the classical solution to this problem is to combine drugs that have distinct mechanisms of action that can be given at efficacious doses and are not cross-resistant. And so, for example, in the perfect world, if you could combine 3 drugs, each of which was associated with a 1 in a million chance of resistance, then the chance that any one cell will be resistant to all 3 is 10 to the minus 18th. And this has worked for tuberculosis, it's worked for AIDS, And it's arguably why we do cure the cancers we do cure, such as lymphoma and testicular cancer. You're making the math work for you rather than against you. And so I'm going to leave you with one slide that, again, I think is another example of a picture being worth more than 1,000 words. This is what the treatment of tuberculosis looked like in the United States. I suspect it looked the same here in the early part of the 20th century, before we started combining active agents for TB. Okay? And I'm old enough to remember what it was like to work in hospitals in the United States in the 80s during the AIDS epidemic, and for a while we feared that we were going to do the same thing all over again with hospitals being full of AIDS patients, until combination chemotherapy was used to control that disease. So I think it's important we remember the power of combination therapy, and to not get overly disheartened when single agents are met with resistance, because I would say it was almost a foregone conclusion that using even good drugs as single agents would be met with resistance. So thank you very much, and thank you for your attention.